All right, it's my really great pleasure to invite Nika Pintar to the stage. She's one of our supporters of the conference, one of our sponsors. So we're really appreciative that you're here. Thank you so much. Whenever you're ready.
Thank you, Morten. First of all, I would like to thank you, Morten, Daniela, and Alex for having us here. It's really a pleasure to be here in this 1 decade of the longevity industry. So hello, everyone. My name is Nika, and I'm the co-founder and chief at Biome. And today, unlike some other lectures, I will not be talking about aging clocks, and I will not be talking about studies on mice, but I will be talking about the effect of fermentation-derived metabolites on real people. Now, 40% of the global population suffers from functional gastrointestinal disorders. 2/3 of those 40% also experience psychological issues, such as anxiety and depression. 40% of them also take medications, leading to really great healthcare costs. But forget about the costs. Think about the opportunities that they are not taking because they are not feeling well. They are not feeling vital. They are not being inspired. Because if you're bloated, if you have to run to the toilet, how could you go to the next level of being? Now, there is something that is very intricate, and that is our relationship to food, because food ultimately truly shapes our gut microbiome. And when we look at food at a molecular level, we have metabolites, which can be anti-inflammatory or pro-inflammatory. At Biome, we created a system that connects both diagnostics and therapeutics in order to detect, predict, and reverse gut inflammation, and ultimately impact our sense of self and vitality, powered by AI. We turned to the world's oldest biotechnology, in which age is a plus, because through the aging process of fermented fluids, we get a wider range of metabolites. And there are several benefits of fermentation. First of all, being that it reduces the content of sugars, that it reduces the content of anti-nutrients. It leads to greater bioavailability of micronutrients that we are all lacking. More than 2 billion people in the world are suffering from micronutrient deficiencies, and they usually don't know about it. And also it leads to lesser content of mycotoxins. So we dig deep. And we also went a step beyond, ensuring that the ingredients that we source are from regenerative agriculture, because we believe that health starts ultimately from the soil that is healthy, that is rich in microbial life, and that leads to greater planetary well-being as well. We designed a multi-level fermentation process that ultimately leads to our H-biotics. They are a biological intervention that includes metabolite microdosing that is very simple to integrate into anyone's daily routine. And they come in the form of a beverage in order to create a total bodily sensation. Because when we have a beverage, it's like drinking tea or coffee. It's something that we experience and can truly integrate into our daily routine. And there is zero technology risk, zero market risk, and zero safety risks because our products are placed on the market as food. They are available today. We already have paying customers. And also what's really important is the lifetime value of our clients, which is 10 times in the ratio towards customer acquisition cost. But in the background, what we are truly building is breakthrough science, which we call metabolites as medicine, where we are understanding the effect of these different synergistic metabolite cocktails on different metabolic pathways in our bodies, and how through the interaction of the gut and the gut cell secretome, we can explore and target different aspects of aging. We conduct extensive metabolomics in collaboration with a lab in Japan that also analyzes extraterrestrial soil for NASA, and we do it at different stages of the fermentation process to truly understand how different plants, herbs, even fermented psilocybin, etc., interact with each other, and what is the result of these interactions. We then divide these metabolites into functional groups, again, to better understand the interplay between them. I will just show you one example of our product development where we can see that the previous generation of H-biotics in comparison to the new generation has a much higher content of acetic acid, which is well known to increase, to support blood sugar levels, and also polyphenols, which are extremely important for many bodily functions. Now, it is my really great honor to also present the results of our first in vitro leaky gut model, where we've seen that already after one dose of H-biotics, there is a significant restoration effect on the barrier integrity, even up to 27%. But besides that, we also measured the effect on anti-inflammatory markers, which also went up significantly. And when we measured the effect on a TNF-alpha, we've seen that there are different responses from the donors, which leads me to my second pillar, which is personalization, because we are all different and we all have different metabolic fingerprints. The way we conduct personalization is through non-invasive diagnostics. We developed our own app, which has 4 different data types integrated into it. First one being dynamic biopsychological assessments. Then we have deep tech biometric scans. Our users can also upload their blood work and other lab data. And we encompass this into one system, also integrating data from wearables to understand the context of the other data points. And when I'm talking about wearables, it's not just the Whoops and the Oura rings, but also CGMs and similar devices. We have more than 90 different data sources that we integrated up to date, and looking for the correlations between them to truly understand and see the effect of our H-biotics on our clients. What we use all this data for is to build the digital gut model. And when our users are first starting their journey with us, we use the following diagnostic criteria, divided into several categories, and that is alteration in gut microbiota, abnormal gut motility, visceral hypersensitivity, psychosocial factors, altered brain gut function, and personality characteristics to truly encompass the gut inflammation and to be able to detect and predict it. But we do not stop there. We also look at the gut cell secretome through the expression and the outcomes in one's psychological well-being, particularly looking at different aspects of stress. I will not be naming all of them, but just some. For example, mood disorders, coping, and quality of life. And then ultimately expressing it in VQ, our Vitaligence quotient. So on one side, we have the data related to metabolites of fermentation. On the other side, we have different types of health-related data from our clients. And in the background, we have multiple machine learning models that all merge into generative Vitaligence to help us build Ani, a virtual being predicting the best biological interventions for us in order to be able to experience peak vitality. However, we do not stop there. We also calibrate our machine learning models and the predictions with extensive scientific trials. I already mentioned the in vitro leaky gut model that has just been finished. We also have ongoing studies where we'll be looking also at the microbial activity, oxidative stress, telomeres, mitochondrial health. We have one human trial in progress already, where we are looking at the effect of the intervention on gut microbiome diversity and inflammatory panel from the blood. And we are already waiting for 2 more ethical approvals for human trials. So this is just part of a much wider scientific strategy, and we are just starting, and we will use the findings to conduct further research. And ultimately, our goal for the next 2 years is to be able to detect, predict, and reverse IBD. And it's based on already conducted in vitro trials which showed, as I mentioned, great therapeutic potential of our H-biotics on restoring the gut barrier integrity, which really correlates significantly to overall health and longevity. Now, we're looking at vitality on a molecular level through the gut in order to build ANI. And I would like to use this opportunity to call every single one of you to join us on this journey, either as an investor or as a new client, a new member, or a scientific collaborator, to support us in the bigger vision in measuring the progress through ANI. Because we can look at the baseline, we can look at the state of where we are now, but ultimately the goal of ANI is to be able to design our unique biology, consequently consciousness, and consequently a completely new world. And we call this world the world of biological transhumanism, where we are able to understand the complex biological processes happening within us, but not only understand, but also design them. So thanks a lot, and approach me afterwards if you'd like to join the Aniverse.
Thank you so much, Nika. We have outlasted one of the projectors here in the meeting, so I guess that's...
I broke it.
Really amazing. Do we have any questions for Nika?
Have you found a way to quantify the permeability of intestinal barrier in humans that is reliable?
No. So that is a good question. So right now what we used is gut-on-a-chip in vitro model to simulate the leaky gut and to see the response. However, what we are doing in our human studies is that we are looking at the effect, for example, at gut microbiome diversity and also inflammatory markers from the blood. And basically, by combining the knowledge from the both through machine learning, we are looking to correlate that and to be able to understand what is happening in humans as well. If you are aware of some new methodology also to explore this in humans, I would like to continue this conversation afterwards.
Okay, thank you so much, Nika. Really amazing.
Thank you.
And thank you so much for the support of the conference. Without you and the other sponsors, it would not be possible. Thank you so much.